Okay.
That's good, Dan. Go ahead.
Okay. So I want to talk a little bit about the Stockholm paradigm, just sort of run through the Stockholm paradigm and the Dhamma protocol again, and adding into this current example we have of warfare ongoing and what some of the implications of that are. And the primary conclusion is that because climate change doesn't take a pause when we decide to have a war, the more expensive, the more extreme climate change becomes, the less capable humanity is of being able to cope with climate change and war at the same time. So we're actually rapidly approaching a point at which war is simply, it's never been a good idea, but it's simply not affordable anymore. And we're seeing this. We saw with COVID that pandemics and climate change are incredibly expensive, and pandemics and climate change and war are simply too expensive for humanity to afford anymore. So, let's see what we're going to do here. All right, there we go. Okay, so the general idea is to remind everybody that the emerging infectious disease crisis is not just a few weird viruses that come out of our tropical rainforest and infect some human beings. That in fact, emerging infectious diseases go from viruses to arthropods. They affect us and they affect all the species that we depend on for survival. So the emerging infectious disease crisis also affects crops and livestock, as well as wild food, plants and animals. It includes diseases we've never seen before, but it also includes diseases that we thought we had contained or eradicated. Emerging infectious diseases are expensive. $1.3 trillion per year is the estimate of treatment costs and production losses for emerging infectious diseases across the planet prior to COVID. So that was the estimate that we had before COVID. And that alone was more than the GDP of all but 15 countries on the planet. So we can understand why adding a COVID pandemic to the already existing emerging infectious disease crisis was enough to crash both the global health system and the global economy. And those costs, in fact, are not just expensive, they are unsustainably expensive. And an article that we just had come out in the last month or so in the Royal Society Open Science Journal includes a very quick modeling exercise that Walter Bieger and the group in Brazil did. And it turns out that if we continue business as usual in terms of ignoring the costs of emerging diseases in food production and just rolling them into increased consumer prices, if we continue that, the costs of emerging infectious diseases in agriculture and livestock production in the US alone will exceed the US gross domestic product by 2080. If we then assume that emerging infectious diseases in human beings are of the same magnitude as EID in agriculture and livestock and food production, then we knock that from 2080 to 2050, when emerging infectious disease costs will exceed the US gross domestic product if we don't change what we're doing. Now, in 1958, Charles Elton wrote a classic book. And in the last chapter, he said that humanity has never faced the magnitude of climate change bearing down on it. And the two major outcomes of that, he predicted, would be conflict and migration. This is a picture of a dozen kids, Ukrainian kids, at the Western train station in Budapest about 4 days ago. These kids are there with two adults, there are other adults around, but this particular group of kids is chaperoned by 2 adult women, probably 2 of the mothers, and all of the rest of the parents stayed behind to fight. And a number of these kids are never going to see their parents again. And if the Russians continue with their current behavior, these children will never even know where their parents' bodies are. And this particular picture from 2 nights ago in the same train station is an illustration of just how complicated the human refugee and migration problem is. This young girl is a Uyghur. She is a Chinese Muslim. She was there with her parents. They obviously had escaped the concentration camps in Western China and gotten to Ukraine, and now they've been bombed out of Ukraine by the Russians. So there are 8 important points that we need to continually pound on policymakers and politicians, as well as our fellow scientists who haven't really been paying attention. The first is that pathogens are not enemies or threats or agents of illness and death. I mean, human beings love drama, but the reality is that pathogens are not any of these dramatic things. They are simply natural components of the biosphere, and their effects on various hosts need to be understood from that perspective. The point of being a pathogen, evolutionarily, is not to cause disease. When an association between a pathogen and a host results in disease, it is an indication that the pathogen is in a host that is marginal fitness space for that pathogen. And one of the things we can infer from that is that there is probably at least one other host where the pathogen is residing quite happily and not causing disease. Number 2, pathogens do not magically appear and disappear. Human beings love magical things, but pathogens are not magical. They do not magically appear and disappear. They are present even when they're not causing disease or making headlines. And we have to understand that when a pandemic is over, it doesn't mean that the pathogen suddenly disappears from the planet. The third is that the emergence of infectious disease does not require new genetic capacities, allowing a pathogen to colonize a new host. The ability to colonize a new host does not have to arise de novo in order for a new host to be colonized. This is the basis of the Stockholm paradigm. Emerging infectious diseases are expressions of pre-existing genetic capacities given new opportunities. That is, the assumption is that there are always susceptible hosts in the biosphere that have not been exposed to a pathogen. If they are exposed to the pathogen will immediately colonize them because the hosts were already susceptible and the pathogens already had the capacity to colonize those hosts. This is what allows diseases to emerge rapidly. And the other thing that's important about this is to remember that new genetic capacities only arise after emergence, that is, after a pathogen has colonized a new host, then new variants appear. And this becomes very important in terms of public policy planning. The fourth is that there's a direct connection between climate change and emerging disease. Now, the health people and the sort of the biodiversity climate change people tend not to talk to each other very well. And so we occasionally, even now, we get we get questions saying, well, how do you know that there's a relationship between climate change and emerging disease? And the answer is that it's a very basic, very simple, but very profoundly important connection. And that is that climate change leads to movement. When climate changes, when climate conditions, environmental conditions change, species living in an area where the environment is changing tend to move away from the altered conditions. When climate change catalyzes species movements, that's what sets the stage for pathogens to come into contact with susceptible hosts they've never seen before. Fifth, agriculture and domestication, trade and travel, urbanization, and war, all human activities since the beginning of the Holocene, create massive new opportunities for emerging infectious diseases. our civilization, our last 15,000 years of becoming a civilized species, have in fact created massive new opportunities for emerging infectious diseases. This is one example from some of Eric Hoberg's work. This involves a simplified picture of the evolution of trichinella, where we see evidence of two different lineages of trichinella originating in domestic pigs in Southeast Asia, where there's evidence of the original domestication of pigs, moving then, presumably being moved by human beings through Eurasia into Europe, where they were then spread to other countries, other continents, as a result of global trade and colonization. Number 6, cities are a problem, not a solution. And we had a punch in the face about that with COVID. I mean, the major places that we had major problems with COVID were big, overcrowded cities. Cities are connectivity and density traps that increase the odds of emerging infectious diseases causing real problems. They are warm. If you want to know what a 3 degrees Celsius world will feel like, just go to any city on this planet in the summer, and you'll know that's what it will be like everywhere. Cities require constant flows of water and material goods, so they're constantly having potential carriers and vectors and agents of disease in and out, in and out. Cities have high population density with low kinship relationships, so there are lots and lots of people, but they don't know each other, and that means that they don't really know who to trust and who to cooperate with. Cities have extreme division of labor and extreme interdependency, which means that even a minor pandemic knocking out, say, 10% of the labor force at any one time can bring an entire city to its knees. Cities provide safe environments for zoonoses by providing wonderful habitats for reservoirs and vectors of disease that find human habitation quite compatible. And every time we create a new urban green space, we create new safe environments for zoonoses to persist within our cities. And cities, big crowded urban cities, maintain a very high quality lifestyle for some people in the cities that are supported by a large number of poor people who are so poor they can't afford to miss work when they're sick. And these people are also generally invisible to public health systems and educational systems. Number 7, emerging infectious diseases do not pause for war. Now, it will be, it probably came as no surprise to everybody that yesterday, all the funding for COVID fell right off the table in budget considerations in the United States Congress for allocating money to help Ukraine try to repel the Russian invasion. This is a big mistake. This is a major mistake to have decided that we no longer had to think about emerging diseases just because we're at war. And this is not new. This is a famous slogan from when I was an undergraduate. This was a slogan that was created by a group called Another Mother for Peace. It was an anti-Vietnam War slogan that war is not healthy for children and other living things. And in 1958, another British scientist, J. Ralph Audi, wrote a seminal article about epidemiology, in which he pointed out that there was enough evidence known by 1958 that it was clear that the geographic range of a pathogen always exceeds the geographic range of disease caused by it. So that means that wherever you see disease popping up associated with a particular pathogen, you can assume that the pathogen actually occurs in more places and is just waiting. So how do we get this kind of silent spread? How do we get a pathogen spread out that far? Well, it turns out that there are two ways to do this. The first is that a pathogen will spread widely through asymptomatic carriers. So people who become infected but are not symptomatic, don't get sick, those people move widely through the environment, or those people, those hosts. As well, when we're dealing with a novel disease, people who do become ill are generally misdiagnosed by clinicians who are not trained to look for novel diseases, and therefore look at a set of symptoms and ascribe those symptoms to something they're already familiar with. So we now know, for example, that in the first 2 months of the COVID pandemic, December 2019, January 2020, before the World Health Organization admitted there was a pandemic, we know now through retrospective studies that a large number of people were dying from what was being termed atypical influenza, which meant they had flu-like symptoms, but they tested negative for influenza, but they died anyway, so we just called it flu. And that's important today because at present, 1/3 of all of the kids crossing the borders from Ukraine into other countries are COVID positive. One of the enormous mistakes in mismanaging the pandemic from the start was that we never had routine widespread testing of children, and children were the primary asymptomatic carriers and reservoirs of COVID, human reservoirs of COVID. We don't know what kind of a risk this presents, but we do know, I know, for example, in Hungary, that as of this past Monday, the masking mandate inside Hungary had been abolished, had been terminated. So no one's wearing masks anymore. But we have 215,000 refugees from Ukraine coming into the country. 1/3 of the kids, they're mostly kids and grandparents, 1/3 of the kids are COVID positive. We don't know what mix of variants they're carrying. We don't know what the impact is going to be. And those people are being moved, of course, they're being moved through Hungary and out into the rest of Europe. And the same thing is happening in Romania and Slovakia and Poland. And we also don't know what else these kids may be bringing. There is a recent study by Gabor Fulvari and some of his colleagues showing that there are 2 hunting tick species, hyalomas, within Hungary that could potentially be suitable vectors for Crimea Congo hemorrhagic fever. And we don't know the risk because we're not looking at this, and because emerging diseases are not important to the policymakers anymore. So nobody's paying attention to the potential for spreading this. We don't know if the Russians, for example, are going to try to release anthrax into the cattle population in Ukraine to try to destroy some of the domestic livestock production. Anthrax also, of course, being capable of getting into human beings. We do not know what's going on, and we're not prepared. We're not looking for it. We're not spending money to monitor these things. We don't know what's going on, and we're not in a position to even think about it. That all sounds pretty negative and pretty horrible, as if the war itself was not bad enough. But there is a bright spot, and the bright spot is that we can actually be proactive about coping with emerging infectious diseases. Okay. And it's based on this very interesting aspect of the Stockholm paradigm. If colonization of a new host only requires new exposure opportunities, given specific pre-existing capacities, that is transmission dynamics, microhabitat preferences, things like that, those pre-existing capacities are predictable. If I say malaria has just shown up in Hungary, everybody listening to this talk is instantly going to say, check the mosquitoes. There's very specific, very specialized pre-existing capacities. So we actually, interestingly enough, we have the capacity scientifically to anticipate disease emergence. We know largely what these things are going to do, how they're going to behave, when they come into contact with a new host. The strategic metaphor for this is anticipate to mitigate. And the assumption is that if we know what's coming, we have a chance to prevent outbreaks or at least mitigate their impact and lower the costs. The tactical metaphor, that is, what do we actually do, is called finding them before they find us, or the DAMA protocol, which is an acronym for document, assess, monitor, and act. The first thing that this requires is that we document pathogen diversity, and we have been terrible at this. There have been calls, large scale calls by the scientific community to document the world's biodiversity for 30 years. And funding has never shown up and never shown up. There's a whole pile of white papers and documents and study documents. There are a lot of plans for how we would do this, but there's virtually no money to do it. And the money that is available is scattered and fragmented, and there's no organization to the actual efforts. And because the money is not widespread and not strategically allocated, what you end up with is groups of people who fight with each other to try to get their particular piece of the pie so that they can do their work. And in order to do that, they have to pretend that the pathogens they're studying are more important than the pathogens anyone else is studying. And that just leads to bad relationships and the inability to cooperate and a very inefficient set of activities by the scientific community. It is important, once we know what we have, it's important to assess the risk. And risk assessment is a three-part process. The first is what we call phylogenetic triage. That is, with modern molecular techniques, we are capable of going into an area and documenting the existence of thousands of pathogens or potential pathogens. We do not have the time to do an extensive, laborious kind of academic exercise on each one of those species. We have to be able to go fast, and we have to decide which ones we need to focus on. So the first thing to do is to place every pathogen that we find into a phylogenetic context, where we can ask two questions. The first is this a known pathogen? And the second is, if it's not a known pathogen, is it a close relative of a known pathogen? Now, if it turns out that the answer is no to both of those questions, then we can ignore that particular pathogen. We archive it, we don't forget about it, but we ignore it in terms of allocating more resources to studying it. If the answer to the first question, is this a known pathogen, is yes, then what we do is notify the relevant public, agriculture, veterinary, or wildlife health authorities that a known problem has popped up in a new place, and they are then capable of dealing with it according to their particular funding and their particular mandate. If, however, we run into something that is not a known pathogen, but it's related to a known pathogen, then we need to really focus on that. We need to assess the risk that it could become a pathogen before it actually shows up in that state. So that requires then that we do what we call phylogenetic assessment. So for species that could potentially become pathogens, we want to know where they originated, we want to know what their original hosts were, and we want to know what their transmission modes are. We want to know how they are actually, how their life cycles are completed. And we would like to know what their likely microhabitat preferences, or what clinicians sometimes call tissue tropisms, where in the body they are likely to live, because that's also a key to what kinds of symptoms they're likely to cause. And then finally, for those particular species, we need to engage in extensive and intensive population genetic and ecological analyses. We need to, and I'll show you how we've done this recently, we need to match fitness profiles for pathogens that we find in hosts where we find them in our inventory projects. We need to match them to a modeling platform to identify likely reservoir hosts and to assess whether or not the pathogen is in a host that we would consider to be marginal fitness space or not. And we need to use ecological niche modeling to determine the likely interfaces for transmission. I'll talk about that in a little bit as well. So here's some work, and what I'm going to tell you is that what I'm about to show you now is the result of work that we did that was initiated by Walter Bieger's group in close collaboration with Valeria Trivolone, when the first sequences of Omicron were published. And it became, in the context of the modeling platform that the Curitiba group have developed, it was really obvious that Omicron was something qualitatively different, not just quantitatively different. Now, in this particular diagram, let me see if I can actually okay. This line here, this is what we would call the business as usual line. So that's sort of the statistical centroid for variants of COVID within human beings. So once COVID got into human beings, all of these variants, and the centroid is this black line, all of those variants are the variants that showed up within the context of infections within human beings. So these are the new variants that show up after humans were colonized. And up here we have Omicron, and Omicron looks really different. If we go down to the bottom here, these diagrams represent sort of a cartoon diagram of the fitness landscape of all these variants. So as you see, you have the initial colonization of humans, and as the variants show up, new variants show up and predominate and go down and switch around, what you find is that basically there's just a variation on this fitness landscape called humans as fitness space for COVID. You get to Omicron and all of a sudden there's this funny outlier. So the human fitness landscape for Omicron is not connected to the human fitness space for the other COVIDs. And this is what told us immediately that Omicron was not the result of something that happened in human beings. It was the result of a human infection going into a non-human host where new variants showed up, one of which we call Omicron, then was capable of infecting human beings again. So this is an example of what we call the oscillation phenomenon or oscillation hypothesis. So when we then do sort of a global phylogenetic analysis of all the records of COVID showing up in non-humans, this is the picture that we get. There are currently an enormous number of COVID variants that are known from non-humans. And many of these have been colonized by infected humans. So it turns out that the business with the idea that Omicron, which is here, might have come from a non-human is not an insane suggestion, given what we know. And the general dynamic is this, where you have something, a bat, for example, and just to sort of oversimplify this, bats to humans in Wuhan, humans to something else, say rodents in South Africa, back to humans. This kind of dynamic, of this oscillating dynamic from one kind of host to another, back and forth, appears to be what is happening with all of these COVID variants that are showing up in showing up in non-humans that are most closely related to COVID variants that are in humans. So we have something that looks like this. So you have a number of variants that are found in non-humans, and all of them arose in humans, and each one of them is capable of then producing new variants, which themselves could then colonize human beings again. And today I noticed there's a new report out today that it appears that some people infected with the Delta variant of COVID, which is down in here, seem to have been co-infected with Omicron, and the two viruses have recombined, and there is now something they're calling Deltacron or something like that. It's a recombinant of the two, and this is entirely predictable. So what do we have in the world today? What we've discovered is that human beings are the primary ecological super spreader of COVID. And almost all of the policy decisions that we made or failed to make with respect to COVID enhance the ability of this virus to spread throughout the world. It is now an entrenched part of pathogen, pathogen pollution throughout the world. Some of you may know that 6 months ago, there was a report of COVID in white-tailed deer in North America. And within the last week or so, there's been the first report of a COVID variant from white-tailed deer infecting human beings. And this is going to happen over and over and over again. It's never going to go away, and it's our own fault. And it's only one example of what we can, human beings can do if they don't understand the evolutionary biology of what they're dealing with. And again, if we go back to the Vietnam War, we find that there is a precedent for this. There was a famous cartoon called Pogo, and this tagline, "We have met the enemy and he is us." This was the person, Walt Kelly, who did that comic strip, said nothing about the Vietnam War for a very long time. And then finally he did a final strip in which this was the end of it. He said, "We have met the enemy and he is us," where he was blaming the United States for the Vietnam War. And of course, he had a lot of trouble with a lot of politicians after that, as you can imagine. But we still keep making the same kind of mistake. The reason the pandemic caused so many problems was because of the way we mismanaged it. And we mismanaged it not because anybody was trying to do something nefarious. We mismanaged it because the people in charge of making public policy are not scientists. They're not evolutionary biologists. They do not understand the basic biology of what they're dealing with. So finally, once we identify the risk that particular potential pathogens pose, those are the ones that are deemed to be most risky are the ones that we monitor most closely. And there are two kinds of things that we need to do in monitoring. The first is that we need to identify the interfaces, the ecological interfaces between the wildlands and managed habitat, farmlands, suburban areas, urban areas, where you have this potential transfer from reservoir hosts to novel hosts that may be diseased. And we need to, in particular, we need to find out what those reservoir hosts are, and when possible, take efforts to mitigate contact between reservoir hosts and susceptible hosts that we care about, like our children, for example. So what we get out of a DAMA project is the ability to act to reduce the risk and therefore the expense of emerging infectious diseases. We find out what's already here, but not yet causing disease, how to reduce exposure to it, knowing the early warning signs of it, how to head it off at the pass. We also gain information about what pathogens might be coming to our places, making it more difficult for them to establish, and also knowing the early warning signs when they inevitably do show up, because climate change is not going to stop and things are going to continue to move. Now, it turns out that DAMA is not just some sort of nice academic exercise that would be an interesting thing to try out if we had enough money, and we never have enough money. The fact of the matter is that if pathogens become established in a new host, they colonize a new host because of predictable pre-existing capacities, that is the point at which we can prevent them. But once they become established in a new host, that's when unpredictable new variants arise. And if we wait until those new variants arise, because they're unpredictable, our only possibility at that point is to engage in unsustainably expensive crisis response, which is what we've been doing and which we already know we can no longer afford. And I would just end by saying Slava Ukraini, and for God's sake, stop this madness. But the question to ask your students from Nebraska is, how many family physicians in the state of Nebraska could diagnose a case of Chagas disease in a Nebraskan who has never traveled outside of the Great Plains?
Yeah, that's really a good question because
armadillos are in Lincoln now. So
yes, running down the street. Yeah. So I think yeah, we don't even know what's right
as
here.
well as we should.
Right
Yeah.
out here.
Yeah.
Yeah, there's marmots that run by here. I can see them going down the ditch, and foxes and skunks. So we have all these things right here in town, right next to Nebraska Hall.
Oh, yeah. Well, you have, I mean, we know that the dogs, cats, and ferrets, we knew from experiments done by a lab in Hong Kong in January of 2020, that dogs, cats, and ferrets were all competent hosts for COVID.
Yeah.
And presumably, and now we know from what's happened in zoos and so on, we have to assume that every canid, every mustelid, every felid on the planet, well, in fact, what we really know now is that because the primary extremely specialized trait that the virus needs to establish in a host, the ACE2 surface protein, that very, very specialized cell surface structure is found in virtually every mammal on the planet. So it's a classic case of an extremely specific but widespread and phylogenetically conservative trait. And that means that the real question is how come we don't have more of these coronavirus outbreaks? And one potential answer that you get from this is sort of accidental literary anthropology. Okay, so there are two of Agatha Christie's novels, her Poirot novels. In two of them, she has a scene in which the detective Poirot is in a doctor's office, and these are one was published in 1932 and one in 1934. And in both cases, there is a bouquet of roses in the doctor's office on the desk. And Poirot says, "What beautiful roses, and they smell so nicely." And the doctor says, "I'll have to take your word for that because I lost my sense of smell permanently 4 years ago when I had the flu."
Very, yes. So,
and if you read her novel called Death on the Nile, there's a description in there. This is something that Eric Hoberg brought to my attention. There's a description in there that sounds very much like a human infection of MERS.
Wow.
So chances are a lot of this stuff has been sort of misdiagnosed because, doctors are trained to treat symptoms. They're not trained to figure out what causes are.
Yes, that's absolutely the truth. I mean, Ethan Jensen said he had a rickettsial disease symptoms that looked like Rocky Mountain spotted fever. He just sent this in a chat. And the physician said, "Well, it can't be that because it doesn't happen here."
Right.
So
it can't be Lyme disease. That's only found in New England.
Yes. And then Sue said, "Many academics and public administration are not able to accommodate contributions from other disciplines because their methods are so narrow, they don't understand anything about what we do. They think science is too far away."
Yeah, no, I mean, that's a really good point. And everybody is guilty of that. I mean, everybody is stovepiped.
Yeah.
And so that's one of the things I've been trying to do in the last 10 years or so in getting myself immersed in science policy and public policy and social sciences kinds of activities, is try to learn techniques for breaking through those stovepipes and getting out to different people. Because ultimately, if we don't cooperate, we're screwed, no matter what we think we know. if we can't put it into action, it's not going to help.